Jean-Philippe Vert. Jean-Philippe Vert is a co-founder and CEO of Bioptimus, an AI-first tech company developing foundation models to drive breakthrough in biology and medicine. In fact, Jean-Philippe Vert has a lot of lives in science. He previously served as a Chief of Research and Development Officer at Hawking, and he was also a research lead at Google Brain. He also led different academic positions at institutions such as École Normale Paris, the Curie Institute, Mines Paris and UC Berkeley. And he's also a member of the National Academy of Technology in France. Jean-Philippe, the floor is yours.
Thank you so much for the opportunity to speak here. Thanks to all the organizers for this fantastic workshop, and you all for waking up to, at the first talk of the last day, I think, which is always the most challenging day in terms of energy. So I'll try to wake you up. I know many topics have been covered this week. I found the program fantastic myself. And I will deep dive a bit today on the use of AI for a specific domain, which is biology. Of course, biology itself is a massive domain. It can go from studying plants to studying medicine, etc. So I'll be a bit focused, but I will try to cover a bit some of the latest development that we and others have done, to share maybe some information to those of you not familiar with the domain, and I'm happy to deep dive on a few topics for the experts in the room. My talk is going to be about foundation models, and maybe to set the stage and motivate a bit why personally I decided to work in that field and devote my time and energy. I focus ultimately myself a lot on applications in healthcare, in medicine, and I think it's fair to say we had a nice conversation yesterday night after the dinner. It's fair to say that we're in a time where technology advances quickly. AI is fantastic. Maybe we'll solve AGI in 6 months, but there are tons of real concrete problems which I think are far from being solved. Some of you are working on climate change and how to do something to positive in that space. I work in healthcare, and in healthcare, health, it's fair to say that today, in spite of all the technology, of all the science, not only AI, but all the discoveries that we've made over the past decades, there are still every year 10 million people dying of cancer in the world, and all projections suggest that it's not going to slow down, quite the opposite. There are tons of diseases. I will not cover all of them. Just one of them that, class of diseases I'm particularly interested in are the so-called rare diseases, which are not very rare at all, because each rare disease does not impact many people, but collectively they impact, I think, more than 300 million people in the world, right? 1 out of 20. And the truth is, for these diseases, for the vast majority of them, for 95% of these diseases, there is currently zero treatment that exists, right? So it's not because we have not searched for them, but something, it means that there is an unmet medical need, right? There are people who will die, who will suffer, and today we don't have a solution for that. So, that motivates why I think some people should spend their energy and brain to try to address these questions. Of course, health is about individual suffering, social pressure. It's also a big economic burden. I need to convince you that today in the world, about 10% of the resources are used to, in hospitals, in drugs, etc., to just try to prevent diseases. So this is also a big economical topic, which explains as well why companies, including ours, can work in that domain, because there is economic potential there to just reduce that cost. Curing better people could also reduce massively the cost of our spending. Okay, so that's the generic, context. Now, you could say, especially if you're not in the field, you could say, well, why don't we make more drugs, right? It seems that once we decide to solve a problem, we should be able to solve it. And so there's an industry, a big industry, with big companies, very smart people, lots of technology, whose job is to make treatments. It's called the pharmaceutical industry, the biotech industry. And so the job of that industry is really to say, well, there's a disease, let's cure it, right? So understand the disease, develop a drug, put it on the market, sell it, and then problem solved. That industry is, by any means, very bad in terms of performance. To put one drug on the market today, we know that it takes more than 10 years. It costs billions of US dollars, so accumulated cost, right? It's very expensive. And to me, the most dramatic statistics is that, it's a very much still today trial and error industry, meaning that if this plot summarizes a bit what it takes to make a drug on the market. It's a long process. I said it takes 10 years. It's typically split into different phases, from the left to the right. On the left, it's mostly basic research. It's about saying you have a disease that has no cure or that has not a good cure. Understand what's going wrong and try to find what it would take to improve the outcome. So understand the biology. Then once you have a hypothesis and a guess on, well, maybe we should target that gene or maybe we should target that pathways. The second step is design a treatment, right? It could be a small molecule, it could be a messenger RNA, it could be a different technology, but design something that will target what you identify first. Then once you have designed it, you test it first in the lab, you test it on cell lines, you test it sometimes on animals. So this collectively takes about 5 to 6 years until you have finally something that you believe will work, because you have understood the disease, you have tested, you have developed and tested your drug, so you're ready to try it on patients, right? This is the step that is called moving from preclinical to clinical research. Clinical is when you first test your drug on patients. And the bad news is that 90% of the drugs that you test on patients end up having no effect or being toxic, meaning they don't end up in drugs. Right? So all the time, all the energy, all the smart things spent understanding disease, developing a drug, testing it on mice, etc., after that, is still at a step where the success rate is only 10%. So this explains a lot why it's so slow, so expensive, because in fact, when I say it costs $2 billion to develop a drug, this is the total spend divided by the number of drugs that come out, and most of the money is spent on drugs that don't work, actually, because it's super expensive. Okay, so obviously, here I'm just telling the problems. One problem is we have diseases that require new drugs, and the second problem is that the people in charge of developing drugs individually are very good, but collectively have a process which does not produce drugs at scale to address the treatments that we need. So a deep question, and this is where I think there's a connection with science, is why are we so good in many things? Like we send we make self-driving cars, we make big machines, we're very good at many things. But why are we so bad at just saying, well, look at that person with take a drug, spend 5 years doing whatever you want to find the best drug you can and test it on patients. Why are we just at 10% success rate? And if there was one generic answer, I think it's just that this is a domain where we have very limited understanding of biology, right? And by biology, I mean the day we have pretty sure, we bet on a drug to try on the patients, it's still a bet. It's still like, well, we hope that the body will behave a bit like we think it should behave, a bit like the cell lines we have used in the lab, a bit like the animal models we have. But there's still a massive jump, a leap of faith. And it's not, it's better than random, but it's still very, very bad. So there is this big fundamental question, which is, could we better understand, what happens in the human body, basically? And this is where I think, this is where I'm, I always try to be optimistic, and I'm super optimistic and bullish on the fact that maybe AI can help on that, right? Because we had debate yesterday about which domains could be impacted by AI, etc. I think recently AI has shown that in domains where we don't have, a few simple equations to describe a system, like language or communication or images, then the recent AI wave brings solutions which we don't really understand. I mean, we have understanding of how AI works, but not perfect, but that turn out to be very, very efficient at capturing regularities, maybe capturing causality, this is a big topic. And so maybe that AI approach could be one solution towards saying, well, maybe we don't know the equation of the human body, but maybe if we use a good AI, we could simulate it, we could generate hypotheses a bit better than what we do today, and maybe move from 10% to 20% success rate, or 50%, right? So that's going to be the topic of my talk, and of course, still super general, but it turns out that biology, I think, is probably so far, we would say the only, but the main success story of AI in science. In 2018, there was this AlphaFold moment, which led to 2 Nobel Prizes in particular, but which really changed the game in one specific domain, which is until 2018, when you had a protein sequence. Protein is a polymer of amino acids, so you can describe a protein as a sequence of building blocks. In a cell, when a protein is created and it falls in 3D, and we know that the 3D shape and the biophysical properties of the proteins are what matters to understand what it does. And until 2018, we were very bad at saying, well, if I take this protein that, for example, I want to create or that I see in a genome, what's its shape? And the AlphaFold answered that question, basically saying, well, now I can train an AI system that takes as input a sequence and outputs the 3-dimensional conformation. And interestingly, there's a kind of problem where, in fact, we know pretty well the equations of physics to describe the structure. So if we had a computer with infinite power, probably we could, from the physical equations, simulate the 3D shape. But this is just, people have tried, and it's just too complex, so we always end up with simplifications that turn out to be not good enough to be useful in terms of coming back to first principles. And the AI approach takes a massive shortcut, which is, let's forget about the physical equations, let's forget about atoms and etc. Just give me enough training examples, millions of sequences, hundreds of thousands of pairs sequence structure, and let's do statistics to try to find correlation between input and output. And it turns out to work very well. Right, so that's the kind of thing where I think AI has already been a game changer in biology, and this technology, and of course, in the following years, many similar and advanced models have been developed to improve it compared to the 2018 model. But this technology today is used in particular in all pharma companies, in all academic labs working with proteins. So massive impact, I would say. Still, and I will explain why, it didn't really change the equation of having 10% success rate in clinical trials, but at least there is hope that AI can move the needle in some domains, and I think biology is one of the main domains where a big needle has been moved. Okay, so thinking of what's in modern AI, what makes it work in LLM, in AlphaFold, I think there is a key component that probably most of you are familiar with, which is the notion of foundation model, which there are different ways to explain it, but overall, I think one of the strengths of this modern AI approach to be efficient is the vision that when you want to solve a problem, it's worth pre-training a model with more data, so in a self-supervised training way, just to prepare a system that understands your domain. So the domain could be human language if you do an LLM, it could be protein sequences if you want to do AlphaFold, but saying pre-training a big model with lots of data can be useful in order to train the system that transforms the domain data into numbers. This can be seen as an encoder of the data. And if you do that well enough, then you can fine-tune or adapt or post-train. There are many ways, but this foundation model for every individual specific tasks. Right? This is what happens in LLMs. LLMs are pre-trained on all the text on the web, and then they are post-trained in different ways to be chatbots, to be to have different functions. Same for models like AlphaFold, even though at the time it was a bit different, but very quickly it converged to that model where to have a good model to simulate proteins, basically the recipe is to say, well, first give me 100 million protein sequences that you can observe in the wild, then show it to a neural network to train a foundation model. And once this is pre-trained, tell me what you want to do. Like, do you want to predict the 3D structure? Do you want to predict the function? Do you want to simulate? All of these are fine-tuning of this foundation model. Okay, so I said this is what LLM does, and the key question here is, okay, how far can we go? Could it work? I hinted that in some domain it already works, like protein folding, but how far could we go into, replacing the much-needed equations for biology by an AI system that could be a foundation model? So at a very high level, the idea is to say, if you have enough data, and then I will go to more details now, in terms of what data are we talking about, how do we represent them, etc. But at high level, if you have enough data, maybe it's possible to pre-train a foundation model that would be a simulator of biology that then you can fine-tune for different purposes, including, I will talk of some example in digital twins, so to simulate, a patient outcome and what outcome in the different scenarios, to make new diagnostics, to help pharma companies find better candidate drugs, etc., etc. Right? Of course, so Bioptimus, the company I work in, is fully focused on that, but we're not the only one. And in fact, in the last few years, and I'm pretty sure that some people in the room may be working on that, and I apologize, I just will mention a few papers out of the hundreds of high-impact publications that appeared in recent years. But many teams and many people have tried, saying, well, if it works, let's try. So if I have images, let's try a foundation model for images. If I have cells, let's try a foundation model for cells. If I have proteins, let's do that. And there's been lots of, I think, very exciting development in that space. So what I'd like to do, quickly now is to review some of the work. So not mine at all, but I think it's useful to, to share with you a little bit some of the recent trends in that space. But maybe before going to specific examples, for those of you not familiar with how it works, foundation model, etc., something quite striking is that not only the idea of saying foundation models, it's just lots of data encoded in a neural network, but also the way to do it is very common and shared across disciplines. And this is also a topic we quickly touched upon yesterday. I said yesterday, maybe it's laziness, maybe it's just because we're in the first phase of testing these ideas, but for sure, in many of the examples that we show, whatever the input data you want to model, it could be protein sequences, it could be cell images, it could be patient trajectories. Today, there's been lots of publication of people saying, let's pick the architecture that works for LLMs and see if it could work for my data. And so that architecture, there are, again, many variants, but overall there is conceptually an idea that what I will show are many methods that learn to model data. And in many cases, the first recipe to model this data is to say, I have some data, so suppose I want to model proteins on the top, or suppose I want to model, at the bottom would be a cell transcriptome. These are very, very different things. But the recipe so far is always to say, well, can I tokenize my data? So tokenization is one of these new words that if you're not familiar with, you should learn. Tokenization is transforming something, a data, into building blocks, right? And so building blocks meaning, so each building block is called a token. And so tokenization is about saying, if I have my data, how can I split it into building blocks and probably encode the relationship between the building blocks? So the best known tokens are in LLMs when you model language, an LLM like GPT, etc. They are encoders of strings. And so tokenization is that you start from a string and you cut it into small pieces, which are not exactly words, which are not exactly letters, but something in between. These are the tokens in LLMs. Well, in biology, people say, well, if I want to model a cell, how can I tokenize a cell? And there's not a unique answer, and I think there is lots of research happening there. But ideas are, well, if I have a cell, maybe the tokens are the genes. So I describe my cell as a collection of individual genes with their expression. And then I have tokenized. I've done the first step. If I have a protein, how can I tokenize that? Again, there are many ideas, but if it's a protein sequence, people say, well, I will describe my tokens as individual amino acids, and then say that the protein is just a sequence of these tokens. Right, and so I could continue the list, but overall, all of the models I will discuss now start by tokenizing data, and then there is, once you have tokenized, you move to the linear algebra space, which is that typically what foundation models do is once you have tokenized your data, meaning you have transformed any input data as a sequence or graph of set of tokens, each token is initially mapped to a vector, fixed dimension. It's called embedding of tokens. And then the goal of this foundation model is to transform this collection of tokens into another vector that describes either each token in its context or the whole, and then can be fine-tuned to do predictions like for a protein, predict a property. Transformers is this machine that go from the input representation of a data as a collection of tokens. So you have already done the job of transforming, let's say here is a DNA sequence into, if you decide that the tokens are individual letters, you describe your sequence as a sequence of vectors. Each vector is an encoding of input token. And transformer is this mathematical operation that transforms this matrix into another matrix. Okay? Therefore, it's called transformer. It transforms. If you open the box, and again, I give you some insight. For some of you, it's probably well known. For others, it's maybe new. So I just want to find a balance to help those of you not familiar with that. But if you say, okay, so what exactly does this? What is the mathematical operation or the computational sequence of operations that the transformer does? So again, transformers are here to transform a matrix of vectors into another one. As input is just a collection of tokens, which each token has some identity, but initially it's just a token by itself. And what we want to do is to transform little by little this representation into another matrix of numbers where the new representation of tokens give the meaning of the token in the context of the other one. So to go from individual and independent tokens to a collective representation of the object. Transformer actually is a sequence of operations. So the transformer is a sequence of more or less identical building blocks, which are called transformer blocks. If you open again the box, inside of each transformer block, there is a lot of operations. But for those of you who are more coming from mathematics, these things are just college level linear algebra operations. They are matrix multiplication. So I won't go to the details, but I think the key idea of transformers, which is not specific to transformer, but which is central to them, is the notion of attention, self-attention, which is to say that in order to, when you have tokens and you want to learn something to change the representation of tokens, first you have a step which is to look at other tokens. And when I say to look at, it's just a mathematical operation. I will show the equation next slide. Just to identify among the other tokens which are the ones which contain information useful to understand what each token of interest is doing. So it's called attention. And then there is a small nonlinear operation to just make sure we can update the representation if we need. So it's very sequential. It's very parallelizable as well, because it's always the same thing that happens. And this is I don't claim that when you see that you understand what happens, but at least there is no mystery in terms of what happens. Right? It's a sequence of matrix multiplication, thresholding. You do that a number of times. And importantly, of course, to state the obvious, all of these operations contain parameters. And the goal of training the foundation model is little by little to update the parameters of the model, right? So that after training, the parameters are fixed and it becomes a deterministic function that transforms input to output. Okay, maybe I'll skip lots of details, but I just to give you an idea as well of where is the innovation when you move from transformers for text to transformers for proteins. There is a space, there is a specific thing in the transformers that caught people's attention, which is the attention. And so I said the attention, mathematically, if you take the basic transformer, is that operation that says when you have, at some step of your successive steps, you have a vector representation for each token. And in the transformer building blocks, I said there is first attention mechanism followed by a nonlinear operations. And the attention here is really, you can represent it as a matrix actually, a matrix of numbers. It's computed automatically, right? So given a set of tokens as input, there is a matrix like that computed that tells you for each token which other tokens are important, right? So mathematically, the numbers here are just a simple operation. It's just matrix multiplication. Each token has two vectors. You do the matrix multiplication, so you get a token per token matrix. But these are numbers intuitively that tell you that to update the vector that represents token t here, then you need to take into account other tokens with a weight. It's a kind of weighted average where the weights are defined by this matrix, right? So that thing is, in the transformer, is learned automatically, meaning when the parameters are learned, they should end up with computing matrices where, if the world are simple, where, for example, the representation of this t may depend on the last a, because if it were biology, maybe the t and the a are close to each other in space, so that's what you want to learn. But technically, it's encoded in this matrix. And interestingly, this is among the many ways to put prior knowledge and to say, well, if I know a little bit about my data, how could I put my knowledge into this model, right? This is a place where there's been lots of effort to put prior knowledge. We had talks yesterday about Bayesian approaches, etc., how to put prior knowledge into statistical modeling. Here, there is very little equation, very little theory, but at least an intuition to say that if a priori you believe that in the attention there is a position that should depend on some other position because you have prior knowledge, then instead of letting the neural network learn it by itself, maybe you can bias the attention numbers to put more weight on what you believe is important. And so this has been done in many different ways. So one of them, and again, there's massive literature on variance of attention. So for example, one of them is called, sorry, to use that, actual attention. Is when your input data, are organized in like a grid. So it has been used in computer vision, but also in protein modeling, because when you model proteins, in some context, it's useful to say, I have one protein sequence, but instead of just looking at the sequence, I have a first step, which is retrieving similar sequences in other organisms to bring lots of context for the protein. So therefore, the input to your encoder, to a transformer, is not only one sequence, but a collection of sequences which are aligned. And then if you don't do anything, you could learn a neural network that says, well, this is just a big matrix of tokens, so I would just learn a big attention system, etc. But that's one case where people say, well, to understand what one letter in one sequence really means, or to learn its biological encoding, I know it's useful to look at the other letters in the same sequence, because a sequence defines a protein. And I know it's useful to look at that position in other species. Probably it's less relevant to look at another position in another species. So if you believe that, then you can start biasing your attention mechanism to look first at columns, then at rows, to tie them, etc. So that, in a sense, you put some constraints in how the attention is computed. And this thing, is one of the ideas that has proved to be successful in terms of, so leading to better models. So that one is typically about reducing the number of parameters. Instead of learning the full matrix and square matrix of weights, you say, well, I just need to learn a vector, a column, a row to do that a small number of times, and by constraining the space of possibilities, turns out that it works in some cases. There are other ideas like that one, which I think has been used also in many variants, which is, your neural network computes a matrix of attention by default, and if you have prior knowledge, that can come from many different sources, but in the case of proteins, for example, you could say, in some cases, you could say, I have a protein sequence which I want to encode, and I have a way, either experimental or because I have another model, to predict what are the physical interactions between possession and 3D. Right? Maybe it's not perfect, but there is a bias that would end up with something that has the same shape and the same size of an attention matrix, but that is not at all attention, which is just 3D proximity. And so if you have these two information, then what people have proposed is to say, well, let's let the neural network learn the attention matrix by itself, but systematically add to the attention matrix my prior 3D proximity score, so that I put a bias in the final attention that tends to be higher in what I believe is important. So you see, that's I just picked these examples as ways that people have explored to put prior knowledge in these models, and therefore, at the end of the day, what we see is that the models used for proteins, for cells, and I will now give a few examples, are very much the same as the LLMs, the same as the core transformers, with a few modifications, and a lot of the modifications so far has been on how you play with the attention. So, meaning that if you want to study that, you need to understand what is the attention, where is it used, etc. Okay, so that was a kind of tutorial and a high-level description of the technology. Now I will just deep dive through a few applications, starting from ones not mine, and then ending with ones we do in our team. But so biology is so large that you say, well, where do I start? And I think it's not chronologically, but one good starting point is to say, well, if I want to learn the language of biology, maybe I should look at DNA, which for biologists is a bit the language of life, right? Say, well, DNA encodes the recipe for life, and then the cell, the proteins, the thing just implement the software written in DNA. So, not surprisingly, people have said, well, let's train an LLM or a foundation model for DNA, so that we can maybe learn DNA just from scratch. And the good news is that there is lots of training data. There is more DNA than text written by humans, right? So potentially it could work. So people have tried that. There's been a succession of attempts, I think in the last 7 years, starting from probably DNA birds, then to other things. But where are we today? I think this is pretty much the latest and largest type of model that has been proposed. So EVO2, which is a typical big model paper with lots of authors from the Arc Institute, Stanford, NVIDIA, etc. So very similar, let's say, to when you want to design an LLM in a big tech company. Just to give you some context about how similar or different is it from LLMs. I think here, so this is a model trained on lots of data. Let's say it's not all DNA that exists, but pretty big portion of it. And in terms of how much is that, that model was trained on 9 trillion tokens. So 9 trillion seems a lot, right? It's 9,000 billions. But all foundation models are trained on lots of tokens. Interestingly, if you look at the state-of-the-art LLMs, GPT, etc., we don't know, I don't know the details of, it's a secret how many tokens they use, but the latest, the ones which are public, say that overall, if you take all the text on the web to pre-train your LLM, you have of the order of 10 trillion tokens, right? So 10, sometimes 15. But so here, in a sense, in number of tokens, this is very, very similar to LLMs, right? The size of the models is a bit below. That model has 40 billion parameters. I think in LLMs, that would be called a smallish model. The big models are hundreds of billions, but similar scale. Okay, so this means that today, the space of LLMs for DNA is very similar in a sense in terms of cost it takes to train, expertise it takes to train, size, etc., to a state-of-the-art LLMs. Is it working? So people have trained it, and then LLMs. You use it every day. I use LLMs every day. Less people use EVO2 every day because maybe it's a bit more cryptic what you do with it. But so if you take the public, here I just comment the publication, right? I didn't do it myself. But the kind of things it has been shown to be quite useful for is first, making sure that some biology has been learned. So one way to see that is to say, well, once you have pre-trained your model, you can apply it to encode a lot of DNA sequences. So encoding means you have a sequence, you have your big transformer, it outputs a vector. So before fine-tuning it for something special, you can look at the vectors and check if it recapitulates the known biology, which is what they did. So they took lots of genomes, encoded them with EVO2 after training, and checked that it describes. I mean, so this is high dimensional. I forgot the size, but maybe we're in dimension 1000. So of course you can visualize that by doing dimension reduction. And there you observe in that space that most of the species are clustered together. So it makes sense. Like if you look at the image, there is nothing shocking, quite the opposite. It seems that just showing 9 trillion tokens to a model, clicking the button learn something that recapitulates what we know about biology, about similarity between species, etc. Then there are more interesting applications. I think it's just the beginning of that domain. So this is before pre-training. But for example, there is a question when you study genomics, for example, in medical genetics, to say, well, we all have slightly different DNA. Most of it is similar, but there are small differences. And it's always interesting, it could be interesting to understand when you see a difference between two persons, A versus T, whether it has an effect. Probably many of the things have very little effect. Some have very strong effects, like predisposition to diseases, etc. And so there's a question: can we use just EVO2 kind of model? So this LLM that has been pre-trained on trillions of tokens from the ocean, etc. Has it learned de novo to predict if a difference in one letter has, for example, an impact on predisposition to a disease? So this can be quantified, like in medicine, we have a list of mutations that we know have an impact, right, predisposition. And interestingly, they checked when you run EVO2 if just using it to encode and something I forgot to say is that these are models that not only encode but are also probabilistic models, meaning you give them a sequence and they can compute the probability of the sequence, right? It's like a sequence. This one is a autoregressive model at the end, a bit like GPT. So what they checked is, okay, give me 2% or the standard human, like average human genome, and the same one with one letter mutated, which could be mine, right? And just compute the probability of the two models. So here there's a big leap of faith saying, well, I compute a probability under a model that is supposed to describe the probability distribution of DNA on Earth. I apply it to my gene A versus Florence gene A that differ by one letter. I will get two probabilities, one more likely than others. And the leap of faith is to say that maybe the probability distribution on Earth tends to favor sequences which are fit to live on Earth, right? So if you have two sequences and one with a larger probability, maybe the one with lower probability has not been favored by evolution and maybe therefore carries more risk for disease, this kind of thing, right? So you can do that, and it's very easy to do it. You take many mutations, you compute the probability and you check if, at least for the human known mutations, which we know are, it's hard to, I don't know which word to use, it's that let's say predisposed to diseases, which maybe in some contexts can be beneficial, but often are not. Then it has been shown that many of these mutations end up with a lower probability for the full sequence under the model. So I think, again, it's still the early stage of this research, but there is signal, and it's quite amazing, I would say, that just pre-training an LLM on all the DNA on Earth captures not only how species are related, but goes to capturing quite a lot about the impact of changes in DNA at the individual level, right? So of course there are many use cases possible. You could think of them being used in the hospitals for genetic counseling, etc. Maybe I'll skip the last one, but of course these models can be used as well to generate new sequences, just like GPT generate new text. You can use that to generate new genomes. Massive bioethical questions, of course, and bioterrorism risk question, but this is supposed to be handled in the paper. But they have some experiments showing that if you prompt the model, there are different ways to prompt, then it can generate sequences that look very much like viable genomes. Right? Okay, so this is DNA. So I would say today there is lots of signs that something has been learned, but we're still very early in, how can we validate, how can we push all of this technology to the real world, like genetic counseling, etc. But it's quite amazing, just to me at least, that just taking an LLM architecture, showing it all the DNA that has been sequenced in the world, captures this thing that seems to recapitulate lots of biology that we have been exploring for years. Going, from DNA to other things, so I will maybe accelerate a bit. Proteins, I mentioned already the story of AlphaFold. This was the first, probably, success story of AI in science. The AlphaFold paper at the time was not presented as pre-train a foundation model and fine-tune it, but when you look at the details, the architecture and also the ways where the data are used is very similar. It starts by collecting lots of sequence only to learn the similarity between sequences and then transforming the many sequences little by little into 3D structure. But anyway, today I think that model is not up to date anymore, right? It was good, but there are much better models today. If you want to look at one recent attempts, again, it's a publication in Science last year by one team in a company. So they developed ESM3, is the name of the model. And here they just say, well, let's take all possible protein sequences we can and pre-train an LLM on them, right? So again, it's a big engineering effort. You need big clusters, you need pre-processing, etc. But it can be done. I think that one is probably one of the largest. So in terms of, again, going to tokens and size of models, we have a little bit less protein sequences than DNA, which we know, right? But we're still not far from the trillion. So 10x less than DNA or LLMs, but still this is the modern science, which is, if you want to train a good model, never start with less than trillion scale tokens, right? Because this is where things start to happen. I skipped the details, but again, this model, and just stating what's in the publication, those things which I find impressive, like here, one of the use case is for protein design. So, not only AlphaFold was, give me a protein sequence and I will predict the structure. Here, the model can be queried to do a bit the reverse, to say, give me a structure or a function that you want, and I will design a sequence that has it. So it's not only generating, but it's conditional generation with a target output. But skipping the detail, they used it, for example, to create new proteins that emit light. So this, if your problem is, I want to design a new protein that, when you put it in some animal model, become green, right? Emits green fluorescence. You can either say this way. I think in the paper they did not go from just a vague NLP to a full protein. They went through a prompting, which is, first you ask a biochemist or an expert what is required to have a green emission in a protein. And this is pretty well understood. This can be quantified as I want my protein to have a shape that allows this thing to happen. Right? So you just describe a specific 3D conformation that you want, and then you click the button and you ask, design me a sequence, so that when it falls in 3D, it contains this pocket that I know will allow emission of green light. Okay? And it's not perfect, but it works pretty well. Like if you do that, then you can generate a first batch of candidates. Some of them are pretty good, but not perfect. And then from the results, you can fine-tune your prompt, redo it a second step. And at the end, they ended up with a few candidates which work very well. So this shows the quick impact of this technology. And again, the pre-training, so the foundation model, was not at all pre-trained for that task. It was just pre-trained to learn the language of protein sequences, but very quickly it's fine-tuned in such applications with state-of-the-art results. Okay, so I will now move to what we do at Bioptimus, which is in the similar space, but as I said initially, what we care is not so much about designing new proteins or understanding DNA of cells, but we would love to, to help people get better treatments and typically understand the biology in humans. So of course there is DNA, there is protein in humans, but there is also, I think the key complexity is the gap between preclinical and clinical, meaning we understand a lot about cells, about simple organisms, but the human body contains probably a level of complexity that is less understood and probably that we can model. So maybe, my dream a bit is building a system. So this is an artistic representation of a transformer, maybe. Think of it as a foundation model, but typically, pre-trained in some way, but then once it's pre-trained, it can be used to input lots of data about individuals or populations, anchor them in some way, and internally have mechanisms to do predictions, simulations, and the range of possibilities, of course, is massive. But for example, if you want to, design new drugs or improve the probability of success in your clinical trials, ideally you would have a machine like that to say, well, I have a drug, I have a patient, my machine should be able, from the patient data, to predict the effect of the drug, and therefore, yes or no, do I go to clinical trial? And if I go to clinical trials, which patients should I, should be the eligible population? Because for many treatments, they work super well on 10% of the population, and they are disastrous on the 90% that remains. So that's the kind of use cases that we work on, and we work that, we build a model and we work with pharma companies and biotechs to test their use, the use in the real world. Of course, this is not a small project, and there are a zillion possibilities to explore. But what I can show as results is, we started actually last year. So the company is 2 years old. So we started last year by a very, less ambitious topic, which is instead of looking at all the patient data, let's start with a few modalities that are easier to collect, etc. We decided to start with one modality, which is images. So this is, I would say, for applications in cancer research, as well as what's called immunoinflammation. And we ask ourselves, can we pre-train a foundation model to be good at reading these images, so that then you can use it for patient stratification or to predict if a patient would respond, etc. Why did we start with that modality? We needed to start somewhere, and I think this modality in particular, so it's called histology, is convenient because it's one of the modalities where you can access large volumes of data, because overall this is, let's say, the standard test that is done when, for example, you have a suspicion of cancer or you have cancer before and after treatment. To be concrete, probably you're all familiar with biopsies. Like if you have a suspicion of cancer, doctors take a biopsy. Biopsy is a small piece of the tumor. And the first thing which is done is you slice the biopsy to get a few millimeters square of piece that you put under the microscope. And this is what you look at. So this is basically used by pathologists to have a first look at the disease, to say whether or not there is a disease, to characterize it, and often it's used to decide the treatment as well. So so therefore it contains lots of information. Information is a bit cryptic because me, even though I look at this image almost every day, I understand nothing, right? I start identifying different types of cells, but it's not easy, I would say. Therefore, there are experts that are trained to do that, but even for experts, we're still discovering. So there is probably lots of information that we don't see, even the experts. And you can. So it's not public data. There is still a complexity. How do you get this data? But this is why, in a company setting, you can find ways to do partnerships, etc., to access this data. So that's what we did. We trained a foundation model, which has a name, Bioptimus. We're still at a scale which is way below the LLMs, I would say. But still, this is today the largest existing model for that modality. 2 billion parameters, so very far away from the 100 billions of state-of-the-art LLMs. And it was pre-trained on 2 billion images. Right? So I did not translate in tokens, but typically we have 250 tokens per image. So in terms of tokens, we're not at a trillion, but we're not too far. And to get that, it's not completely easy, but this is obtained from a few millions of patients across many hospitals. Right? So this is already something that a single hospital cannot do, a single team cannot do. It's data aggregation is required, but this is what we managed to do. Is it working well? So actually, in this modality, we are not the only one to work. So it's very cool to be competitive domain where every 6 months you have a new model. Our model seems to be working very well. Like it's number one in many benchmarks. This is one benchmark published by University of Hong Kong of Science and Technology recently. Still online. So I checked yesterday. We are still number one. Importantly, this is a benchmark very similar to the benchmarks you see in LLMs when you can compare Claude and GPT on their ability to solve math problems, etc. Here it's about how good the encoders for histopathology images are when you fine-tune them for clinically relevant tasks, which are about, can you predict the outcome of the disease? Can you predict the response to a treatment? Can you predict a genetic mutation from the image, etc., etc. So there's a long list. Actually, there are 229 tasks in that benchmark. And there are differences between the models. I won't go to details of individual tasks, but in terms of absolute numbers, so this is another benchmark. What I find interesting is that the value, so the numbers, the performance that you get, has been improving a lot since people have started doing foundation models. So here, this is again an average on many tasks. Typically, you have a breast cancer image and you need to decide the risk of relapse, right? This is one task. So you can formalize it, you get some AUC. About 5 years ago, before foundation model, people were doing computer vision systems, CNNs, etc. And they were about at 75% accuracy on that benchmarks. Today, we jumped to 85. So in just a few years, I'm not sure, people like the press will call that the AlphaFold moment, but there is steady improvement. And, going from 75 to 85, and there is no reason to believe it's stopping, saves a large number of lives, right? Because these numbers, when you turn them into diagnostic systems, into medical decisions, ends up taking better decisions. So something is happening and across many tasks and all the studies. So I mentioned just a few numbers here, but go to the direction of saying, well, it's a fact that if you can get more data, bigger models, train the right model, then you see improvement across the board when you fine-tune the model on many tasks. Right. So good news. The foundation model ID seems to be one way to bring improvement in these domains. To be concrete, we not only we go to benchmarks, but we also work with partners to say, okay, it works well, so turn it into something that would impact patients directly. It's still at the research phase, but we've been working with academic group at ICGI, which is the Oslo hospital, on using this model in order to, so that one is to help pathologists automatically do a report of what they see on the image. Right? So in a sense, it's something that is well known in the tech industry. You have an image and you want to write a text to describe what's inside. Here we want to do the same, but it's a histopathology image, and you want to write the medical report. And they won a challenge recently using our encoder for the image and then LLM to write the report. So this shows that we're very close to turning these models into in the softwares used by pathologists that will accelerate their work. We also work with MIT, with the lab of Regina Barzelay, on using these systems to predict the risk of relapse in breast cancer. Again, they won a competition using it. So, it's coming together, and the next step is to transform that into a diagnostic system for the patients. Okay, so that was step one. Can we train a foundation model on one modality and start having a system that captures information, summarizes it, and does right prediction? But it's just the beginning. Remember, I want to input all modalities. So we're not there yet. We're working on that, but we already have a few steps in the right direction, I think, which is, okay, if you have an image, can you not only give overall predictions about patients, but could it be that there is information to predict or understand what biology is happening inside of the image, like in each cell, what genes are expressed, what proteins are expressed. So this can be formalized, a bit as a machine learning problem, which is modality to modality translation. You have an image you want to translate into the space of, from pixels to gene expression, for example. We worked on that, so there was a newspaper last year, not from us, again, independent work, that did a benchmark on that. We have a model that works well, but interestingly, the performance, this is an average performance. So here the question is, from the image, can you predict the expression of genes across the image? Okay. Interestingly, there are some genes for which us and others as well, but we have very, very accurate prediction. So sometimes we have 90% accuracy between the prediction of where the gene is expressed and the ground truth, which you can obtain if you're ready to pay $10,000 per sample to get the expression of the genes. There are other genes for which the signal is much weaker. I think there's going to be progress, with better model where we can improve more genes. But across all the genes, there are many ones for which the prediction is high enough to anticipate that today you could take just these H&E images, so the very cheap ones, and you can get millions of them, and systematically augment them now with in silico prediction of gene expression, which suddenly allow people, clinicians to do studies like, okay, now that I have my patients, not only the image, but many genes, can I find correlation between the activity of genes and some clinical outcome that could help me go back to the biology, maybe find new targets, etc. So this is one of the very exciting activities going on. Just one example, the image is bad, but what we can do today is you take just an H&E image. As I said, there are some genes you can predict well. One of them is called CD28. Turns out to be a gene which is known to be super important to predict patient response to immunotherapy. But the way it's known to be important is that if it's expressed in one type of cell, which are T cells, so immune cells, it's good. If it's expressed in cancer cells, it's bad in terms of response. And so what we can do today is just from an image to predict cancer cells, T cells, and to predict the expression of the gene, and to overlap all of that to suddenly create new biomarkers that transform an image like that, implement what we know about the likelihood of response, and run that on millions of patients. So coming back to historical data and check if indeed there is a good correlation between these in silico computed gene expression and the response. So very exciting research. I know I'm on time, so we just finished with 3 questions, which I'd love you and the community to work on, because there are so many things ahead of us. So one is, we're just at the beginning. I said we started training some models on one modality, 2 modalities. Now we're raising soon a new model. But the space of possibilities is wide. Like, what's the right architecture? What's the right tokenization? What's the right objective function when you train the model, etc. We have some ideas, but this is really, this is very, very open. Second question, causality. Crucial question, because of course many of these models, I say, well, we want to encode the knowledge to make predictions and ideally to do counterfactuals or simulations under different scenarios. There are many people working on that. There are key questions, but bottom line is it's hard because when you have patient data, you have very little interventional data. You can have lots of intervention in preclinical model, in cell lines, etc. But so there is a big topic: how can you capture causal signal and make sure that your system eventually will be good enough to predict patient trajectory under different scenarios? And finally, I know there was a day I was not here, but on agents, one of the very active domains where also we work on and we have lots of interactions with other companies is that if we manage to train this machine, which is kind of one model of biology, right? It can read data, it can do simulation, then we see how humans can use it. And so far, we mostly ask humans to use it, like a doctor, make a test, etc. But tomorrow, it's very clear, today and tomorrow, it's very clear that agents can navigate the space and accelerate this progression. So I think this is also why these foundation models are useful, not only to be fine-tuned by humans, but now to give the environment that these agents will learn to interact with. Thank you for your attention, and I don't know if you can take questions, but I'll be here today. Thank you.
Thank you for the brief talk. So what we are going to do, we are going to take 2 questions in the interest of time, and I wish I ask you to take the coffee break opportunity also to ask questions, because we have also the next talk is online. But yes, please go ahead.
Hi. Thanks for the talk. So how are we in the state of distilling these models in order to put them into production or even into edge devices?
It depends. So the histology model, like the ones we've developed, for example, ourselves, we already have pre-trained a big model and distilled them. Not so much to put them in edge, in your smartphone, etc. Maybe that could be a use case, but so far our use cases are more in the hospitals. But also it's useful in terms of cost, of course, inference. I forgot to say, these images are gigantic. So, when I say we have a 1 billion parameter model, it's just to encode a 224 by 224 sub-image. So the cost to encode a big image can be quite expensive. So we already have distilled. And distillation works quite well. A bit like LLMs, we see so distillation, right? It's taking a big model and making it, training a small model to imitate the big ones, more or less. Our experiments show that we just like for many other distillation cases, we keep more or less the accuracy, but can train 100 million model parameters from 1 billion, for example.
Great talk. Thank you very much. You showed a bunch of different modalities that you'd be looking at in the future. Do you then envision having multiple small distilled models that are experts at the particular modality, and how do you plan on combining them all? Ask one question, have all models give you the answer.
So this is one of my questions. I don't have. I think it's widely open. Our approach for the moment is to jointly train one big model from all modalities, but from data which can be aligned across modalities. Like you have patients with both an image and genome, and also data which are isolated, so just images, just genomes, etc. But so far, our approach is to have a single model to do that. We also explore variants where you pre-train individual models and then connect them. But frankly, this is we'll see in 10 years what's the approach that works well.
Okay, thank you very much for the interest of time.